Hello all, welcome to another CSIR Net previous year question series from Life Science Library. Now we are going to see part 2. Drosophila CSIR Net previous year questions from 2008 to 2017. It will be useful for CSIR Net GATE and Life Science candidates. So far we have seen all these questions in our previous lecture till 2017. Till this question we have seen yesterday. Today we are going to see about another question from GATE 2018, Life Science GATE Life Science. So what will happen if ultra-bithyrax is overexpressed? If ultra-bithyrax is overexpressed, ultra-bithyrax refers to T3, that is formation of legs and halters. It will form flies with 4 halters. C is the right answer. Now from Drosophila, a segmentation question from December 2015. So hedgehog, by seeing these options we can see hedgehog is not a transcription factor. D is not right. We can eliminate this. Engrilled is secretory factor. Engrilled is not secretory factor. Engrilled will secrete hedgehog. So E is not right answer. Arriving at answer A and B. Expression of wingless and engrilled is activated by pair rule genes. Continued expression of wingless and engrilled are maintained by interaction between these two cells expressing engrilled and wingless. B. D is the right answer. We are aiming for correct options. So D is the right answer. Next about wingless interference RNA. If the wingless interference RNA is formed, it will repress the function of wingless. Since wingless proteins make long range gradient, no, wingless protein will not make long range gradient. B is wrong answer. Bilkul galat. Next, enhanced expression of wingless. We are introducing interference RNA. So, expression of wingless will not be enhanced. A is not right answer. Since engrilled expression is initiated by pair rule genes, posterior segment will not be formed. We are speaking only about wingless. So, we have nothing to do with. Yes, wingless. The concept here is consider wingless. This has wingless gene, this has engrilled. Wingless, engrilled. Wingless will secrete wingless protein. Secretory factor. It will go and attach engrilled and it will stimulate engrilled to form frizzled receptor. It forms patched receptor. It forms hedgehog. Go and attach hedgehog. So this is what the process. If engrilled is not functioning, it will interfere with wingless is not function. So engrilled will be affected. This is anterior segment, this is posterior segment. So engrilled will be affected. Right answer is C is the right answer. June 2015. Okay. Then December 2014. C. Mutant experimentally generated to reduce wings to halters. Okay. This is what given in the question. Following are the statement put forth. We are going to find the correct statement. That is ultra-bithyrax is expressed. That is T2 is made as ultra-bithyrax. Wings are reduced to halters. So then, reduced to halter-like structure. So Antennapediya codes for wing. B is not right answer. B is galat. Homeotic mutation is correct. Ultra-bithyrax codes for halters. So halters are overexpressed. A is correct. A and C are the right answer. Now a question from June 2014. Torpedo and Gherkin concept. Torpedo is a transmembrane receptor on follicle cell that binds with Gherkin protein located in the presumptive dorsal surface of oocytes. and inhibits a cascade leading to the neutral localization of dorsal protein. In an experiment, Drosophila germline chimeras were made by interchanging the pole cells of germline precursors between wild type embryo and the embryo from mother's zygote for mutation. Consider the two statements. Read the two statements carefully. Wild type female. Wild type female. Okay. Whose egg come from mutant mother. Torpedo deficient females. Whose egg come from wild type mother. These are the two statements given. You are going to find correct option. Okay. First statement. Torpedo deficient eggs develop in the wild type ovary. Produce normal embryos. Correct. Wild type eggs developed in the torpedo deficient ovary produce ventralized embryo. Okay. Correct. Torpedo deficient eggs developed in wild type ovary produced ventralized embryo. No, the egg is torpedo deficient, but ovary is wild type. So it will be normal. It will not form ventralized embryo. C is not right. Dorsal proteins enter into the nuclei of dorsal side embryo, which came from the wild type eggs and developed in torpedo. This is irrelevant statement. Dorsal protein remain in cytoplasm in the dorsal side of embryo, which came from wild type eggs and developed in torpedo deficient ovary. See, now we are going to study about dorsal protein. What is the duty of dorsal protein? Dorsal protein, once detached from dorsal protein, will be in complex with cactus. If cactus are phosphorylated, this cactus will be degraded and dorsal protein will make an entry into nucleus. When dorsal protein enters into nucleus, it forms ventral. This is the concept of dorsal protein. So now let's read again. Dorsal protein enters in the nucleus of the dorsal side of the embryo, which came from the wild type eggs developed in the torpedo deficient ovary, because the ovary is torpedo deficient. The ovary itself is torpedo deficient and the egg can only produce gherkin. Ovary can produce torpedo. If the torpedo is deficient, so the egg will be ventralized. So this is right. A is right. Egg protein remains in the cytoplasm in the dorsal side of the embryo, which came from the wild type and developed in the torpedo deficient ovary. This statement is not fully completed because dorsal protein remain in the cytoplasm in the dorsal side of the embryo which came. It will not remain in the cytoplasm. It will enter into nucleus, no? So E can't be correct. A is the right answer. Now, Drosophila, again discs. Change in the state of specification of the imaginal disc in the Drosophila that of a different disc type is known as transdetermination. Changing the determination. Determination is nothing but most important terminology in developmental biology. We have seen in previous video that if a cell is determined to give rise to I, consider. It will not be changed any time in the life to form different organ. This is called as determination. If that is changed to that of different, change in the state of specification is known as transdetermination. Okay. Another question from French Plague Analogy, June 2013. Transplanted cell retained their committed state. So what is the question? In the experiment, the cells could normally become the middle segment of the Drosophila egg were removed from the leg and they are placed in the tip of the antenna of the fly. What will happen? It will form fusion structure. Fusion structure of retain their committed status of leg plus antenna. They form claw-like structure. A is the right answer. Homologue of beta-catenin in Drosophila is armadillo. Drosophila mutation in homeotic gene results in which one of the following developmental defect? Segment-specific identities will be lost. Anterior structures in the Drosophila embryo. So, anterior structure with reference to concepts. Anterior structure will be associated with bicoid and hunchback. Two things associated with anterior segment. Posterior segment associated with nanos and caudal. The posterior segment. So B is the right answer. Zygotic gene is required for the adjacent segment of the Drosophila will refers to gap gene. Straightforward. DV axis, dorsal ventral axis, is maintained by sonic hedgehog. December 2020. Dorsal, so now I said dorsal protein will enter into nucleus and will form ventral fate. A is the right answer. If, so I said now dorsal protein will be attached with cactus. If cactus is phosphorylated, it will be degraded. So this dorsal protein will enter into nucleus. If this dorsal protein enter into nucleus, they transcribe and translate, and it will give rise to ventral fate. This is what the actual concept. Now this question is asking if the dorsal protein is mutant, what will happen? Ventral fate will not be formed and the entire egg will be dorsalized. Okay. Yeah, 2008. Dorsal mutated in Drosophila is a result of, if dorsal is mutant, then the egg will be dorsalized. A is the right answer. So far we have seen almost all the questions except one question. Question number 156, June 2013. Take it as homework. Segmentation genes in Drosophila are divided into three groups: gap gene, parallel gene and segment polarity gene based on the mutant type. Below are some of the major genes expressed in a sequential manner with respect to groups affecting the segmentation pattern. So we know bicoid and hunchback refers to anterior segment, and the two things, that is wingless and engrilled. Engrilled will be produced with high concentration of even skipped fusitteraso and paired. Engrilled will have high expression of these three things. Hunchback, even skipped fusitteraso and wingless. Tailless, hairy fusitteraso and gooseberry. T is the right answer. Hunchback will induce even skipped fusitteraso, wingless. Tailless, hairy fusitteraso give rise to gooseberry. So think that Hunchback is anterior segment. Even skipped Fusitaraso is posterior segment. So this hunchback will go and induce this. It will have an even segment, even skipped in Fusitaraso. It will produce hedgehog signaling. It will again stimulate wingless. This is one kind of signaling. Other time tailless. Tailless is posterior segment. It induces hairy Fusitaraso in it. They are stimulated by wingless. So again, hairy and Fusitaraso are stimulated. Gooseberry. Gooseberry is dorsal. It gives rise to dorsal protein. So it is a segmentation topic in Drosophila. These are the concepts. There are three things: gap gene, parallel gene, and segment gap gene, parallel gene, segment polarity gene, and homeotic gene. This engrilled Fusitaraso, even skipped, paired, sloppy paired, all this comes under segment polarity. This is the concept. Much more conceptual clarity, please comment in the comment box below and take it as a homework, explaining all those things in the usual clear-cut manner what we do in our channel every time. Try explaining this question in comments. We'll be taking this in next video. Hope this video series is very useful for preparing CSIR NET GATE Life Science for the topic of Drosophila Drosophila topic. Hope if you think this video is very useful, please do refer this channel with your friends and fellow aspirants. Please do like, share and subscribe to my channel.